A sourdough starter is very easy to work with and one of the healthiest breads to make. As a start, you only need water, flour, and thyme to make one. The flour and water are mixed, and you feed them every 24 hours with more flour and water. After a few days, you should see it start to rise and fall. This is because of the metabolism of various microbes that grew from the flour, including lactobacilli, yeast, and other bacteria that are found in wine, vinegar, beer, and other foods that come from fermentation. Many types of microbes convert different sugars and molecules from flour into acids, carbon dioxide, and water. This acid is what contributes to the taste of sourdough bread. The carbon dioxide waste is also what causes the starter to rise, as the newly formed gas is trapped in the dough. My personal big question was about comparing people's control groups to their experimental group in regards to how their cultures developed. Are all the strains the same in each of the cultures? Is the demographic of the microhabitat of the starter the same in regards to the dominant microbe species? Are there any microbes that are present in one but not the other? Each person in a group of over 150 had at least 2 starters, 1 control, and at least 1 experimental. In the experimental groups, people put many kinds of fruit in them, although there was a limit of 1 fruit to each experimental group. In the control group, there was just water mixed with flour. My personal experiments did not go so well, as both groups formed a hooch, which is a liquid consisting mostly of alcohol that forms because of conditions that are not ideal for microbial growth. I did not know that at the time, and my cultures mostly died a few days afterward. To find the different species of microbes that were present in the starters, we had to use DNA sequencing for the entire culture. This included looking at a single gene in bacteria in a certain region in the genome in fungi, the fungi being the yeast used in making the sourdough. There are 2 types of sequencing: shotgun sequencing and amplicon-based sequencing. Shotgun sequencing is looking at the entire metagenome, or all the genetic material of all the microbes in the starter, while next-generation amplicon sequencing just looks at a few genes like the one in bacteria and fungi that this particular study is looking at. With amplicon-based sequencing, the first thing to do is destroy the cells and remove the material except for the DNA. Then, put the DNA in tubes and clone them using a polymerase chain reaction where you use an enzyme to make copies of the DNA over and over and over again of certain genes. Then you create barcodes made of nucleotides that are unique to each tube. By looking at each region of a given unknown bacterium, you can identify its species and what tube it belongs to. This is how you get the species of the microbes in each sample of a starter. The original question I was going to ask was, does each smell correlate with a specific culture? However, this was difficult in answering because looking at the data of aromas versus orders of organisms of 2020's fall semester, many put down their aromas as not applicable. This could mean many things, such as there wasn't any smell, it smelled like flour, or the smell was indescribable. In addition, some microbes had a major presence in samples with a smell, but negligible presence in samples marked with not applicable. As a result, I refined my question to be, does everyone have experimental cultures similar to their control cultures? To find out, I studied 2 samples from this semester by Anna Nelson. She used blueberries in hers, and for the most part, the answer to my question in reference to hers was yes, the experimental group's demographics were similar to the control group. However, one major difference was that in the experimental group, there was a relatively large population of Acidobacter, which had little to no presence in the control group. When researching Acidobacter, I found out that it is used to make beer, vinegar, and wine. It converts ethanol, which is an alcohol made by decomposing sugars, into acetic acid. Then the bacteria converts the acid to carbon dioxide and water. Converting the ethanol to an acid causes the experiment's pH to go down, and the bacteria has genes to protect itself from the acid, such as the production of citrate. With this in mind, I hypothesize that this bacterium will be found in already rotting sweet fruit due to the sugar content, and sour fruit due to the acid content.